All right, it's my pleasure to introduce Jonah. He's a PhD student at Mississippi State University, working with Dr. Graham Rosser and Dr. Matt Griffin. His PhD project focuses on parasites of the invasive apple snails, especially the rat lungworm. But he also likes to research understudied fish parasites, and I'm sure he's having a great time in Graham's lab. Take it away, Jonah.
Hey, yeah, so this is going to be more of like a overview of my project. I've only done, a very small portion of it so far. So it's not going to be much of like a uh intro methods results type thing. It's going to be more of just like a little interesting overview of what I'm doing and some info on rat lungworm and stuff like that. And of course, this isn't even actually about fish technically, but it is about snails. So these are aquatic snails, so kind of related. So some background on rat lungworm to begin with. Pretty important. So this is rat lungworm on the right there, you can see, and its definitive host, so where it sexually matures, is in rats. It requires an intermediate host to complete its life cycle. In this case, for this parasite, it's gastropods, snails and slugs. It has many paratinic hosts, even more than the ones named there. But paratinic hosts basically are kind of like transport hosts, or maybe kind of accidental hosts, kind of, where the parasite gets inside of them but doesn't develop. But it's if another host were to consume that paratinic host, life cycle could continue. Um, and that's kind of where my project comes in, looking at that a little bit. And then aberrant hosts, kind of a similar thing to paratinic hosts in a way, but think of it kind of as like when a host that's not supposed to be the definitive host um, gets infected. So like you're kind of taking the place of the rat in this case. And that's where a lot of the problems happen. Paratinic hosts don't have that many clinical signs, don't have that many issues, but aberrant hosts, when it gets in something other than a rat, for that particular life cycle or life cycle stage of the nematode, then there can be a lot of issues, and that's kind of where my stuff comes into also. And when it's in the aberrant host, when it's inside of us, for example, it goes to the central nervous system a lot for some reason. Occasionally lungs and eyes and stuff, but central nervous system, it goes to a lot in aberrant hosts. So that's kind of the reason why it's important. And then the distribution, it's all over the world, but technically, in each country, but within the US, it's concentrated, especially in Hawaii in the southeast US. So just quick life cycle. It's in rats. Um, they get shed and then ingested by a gastropod. It matures to an L3 stage in the gastropod. And then the gastropod either like sheds slime onto produce, and that's ingested by humans. So this contaminated produce is ingested by humans, or a paratinic host ingests this L3, and then a human ingests the L3 within the paratinic host. So kind of complicated. The point is the human ingests an L3 developmental stage in some way, and that's how they get infected. They couldn't just eat a rat and get infected. It would have to be at an L3 stage. Anyway, kind of technical, not really that important actually for this discussion, but pathogenesis, briefly, yeah, and it causes eosinophilic meningitis. A lot of time though, it's self-limiting, doesn't cause much issues, but the upper end of what can happen, which includes paralysis and death, is pretty high. So it's a pretty big deal. To rats, it does nothing pretty much. So of course. Just more interesting kind of wow factor stuff here. So this is a human brain here, and you can see this is a rat lungworm larvae embedded in the brain there. There's some lungs it was in from, I think, like a 3-year-old boy, which is pretty depressing. That caused like infarctions and stuff like that and issues with their lungs. And of course, like a lot of headlines, like you might have heard of this, like the Australian man who dared to eat a slug got paralysis and died after 8 years. Like you might have heard of that. That's caused by rat lungworm. He ate a slug and then sadly got paralysis and died years later. So this is more just to show like a sad dog. Dogs, when they get it, they get hind limb paralysis, and especially. So that happens a lot in Hawaii. But it could happen to dogs here too if your dog were to ingest a slug or a snail with rat lungworm. Anyway, prevention, if you want to prevent this, just like if you want to cook if you want to eat snails or slugs, you can technically. Same with like Pertinicoast. If you want to eat like a lizard or something caught in the wild, of course you could. I'd probably recommend not doing it just in case, but you can cook to 165. I mean, it'll kill rat lungworm. In fact, you could technically cook less than that based off research. The L3s die at a lower temperature than that, but who wants to cut it that close? Like, just cook your food. In fact, just don't eat it at all. Probably, it's what you should do. Anyway, so moving on to research goals, it's mostly just to characterize parasites of apple snails in the southeast. So trematodes in the form of cercaria shedding from the snails into the water, nematodes, things like that. And then aim two, the bigger one especially, is to determine the prevalence of rat lungworm in apple snails in the southeast, especially in states immediately around Louisiana, because the concern is that there's apple snails in crawfish ponds in Louisiana, and they're there. They're in some of the crawfish ponds in Louisiana. Apple snails can get rat lungworm. We know that already. Could the crawfish serve as parotinic hosts by consuming dead apple snails that have rat lungworm? That's the idea. And then could people get infected by eating those crawfish? So a huge chain. So an update on the project right now. I got to go fast. I don't got much time left, but I've necropsied like 27 or so snails. It takes a long time. Doesn't sound like a lot. It takes like 3 hours per snail. So that's more of like the general parasitology breakdown of what's inside of the snails. I found a lot of nematodes in the stomach and intestine. Haven't sequenced most of them yet, but most of them are probably based off the morphology, just like plant parasitic or free-living nematodes that got consumed by the apple snail, so not really that important. I found an encysted nematode in one that's probably parasitic. It was encysted in the snail, so parasite of the apple snail. Who knows what it infects next? I need to molecular ID it. I'll go over all these, but I found a lot of putative cysts, so like they look like cysts of parasites. And then a lot of notable sequencing findings. So I'll just jump right in. This is a species of apple snail, its distribution currently. This is just one species of Pamacia, but this is, you can see where it's distributed across the US. These are my sampling locations so far, so not too much. I could use more. This is a bunch of apple snails I've collected just as an example. You can see what they look like. They're rather large. That's why they're called apple snails. And they lay these pink eggs that are really weird, interesting to look at. So pretty cool. These are some examples of intestinal nematodes I found. As you can see, yeah, they look like nematodes. I haven't done too much molecular work with them yet, but the sizes for them, some of them are just way off, way too small or way too large to be rat lungworm. So ruled out, can't be rat lungworm. Some of the morphology also is just off with other things people use to ID nematodes, but just some interesting examples of what I found. This is an example of one I've sequenced. This picture is pretty terrible, but I've gotten better at taking pictures since then. But the COX-1 sequencing just showed it being very distant to anything else in GenBank, like usual, when you're working with obscure things. But its closest relatives are plant parasites or free-living nematodes. So probably, like I said, just free-living or plant parasitic nematode just passing on its way through the apple snail. This is where things get more interesting. Oh, these are supposed to be playing. That's fine. There was a cyst in here, and I cut it open, and a nematode came out, a rather large one, way too large to be rat lungworm, so not that. So interesting also. This is definitely a parasitic nematode of the apple snail at the very least, and probably something else consumes the apple snail and would get infected. You can kind of see it off to the right here. There's supposed to be a video, but that's fine. Oh, there you go. Now it's playing. I had to click the arrow. So pretty cool, right? Many putative cysts that just need a molecular ID. These were frozen, so I couldn't just like pop open the cyst and see what was inside of it. It's frozen tissue, so it is what it is. This is an example of another parasite I found. Cephalin. It's this one, super long name. And the life cycle of this is already known. Turtles consume the snails and become infected. So this would infect a turtle if a turtle were to consume an apple snail. Pretty cool. I have found an encysted trematode, kind of boring. It was one of those putative cysts from back there. Anyway, I sequenced it. Closest relative is to ones that infect marine or freshwater fish, so kind of cool. It just shows that, yeah, these things can have cysts that fish, they can have cysts of things that infect fish, probably. So kind of interesting. It just shows that there's some good ideas going on in the project. This is the more interesting thing, probably. So these are cercaria shed from an apple snail I found, all in the last day of that petri dish, all Cercaria. Crazy. In fact, that's only a small fraction of the ones shed from this apple snail. So the amount of Cercaria, that is like larval trematodes pumped out in the water, crazy high by these apple snails. You can see them squiggling around here. But I've done sequencing. It's not close to pretty much anything. Besides for this stuff down here, and some of those things are zoonotic. So these, it's probably not zoonotic, but it's related to things that are. So there's some risks there. I found that also in Panama City Beach. Is it the same thing? It looks very similar. They are the same thing. So sorry I'm blowing through this fast. Too much content to go here. But they are the same thing. These two Cercaria from Panama City Beach and Mobile. So I found the same thing in both locations, but it makes sense since they're so close together, Mobile and Panama City Beach. I'm just going to blow through that. So the future plans especially are to perform qPCRs on apple snail tissue that I have saved in the freezer, so we can get a prevalence rate of rat lungworm via qPCR. And that's where you could come in. You could send me apple snails, maybe. So maybe save my contact information if you're interested. Really only interested in the Southeast US. So yeah, that's about it. Sorry I had to blow through a lot of stuff, but too big of a presentation.
Yeah, all good. That was interesting project, Jonah. Curious to see what you find in more snails and when you start looking at the crayfish. Folks, if you've got a question, feel free to drop it into the chat or just unmute yourself. I'll ask you one, Jonah. Have you found anything that seems to be pathogenic to the snails while you're looking at them?
That's a hard question to answer. Cercaria, when they're in high enough amounts, can castrate the snail and cause them not to be able to reproduce. So in that way, you could say so. To know more, we'd have to do histopathology, which you can do on snails. So we'll probably end up doing that eventually. But honestly, we don't really care about the snails that much because we're not looking to like weaponize parasites to kill apple snails. So that would kind of be more of like a different project. But yeah, we will investigate it a little bit, probably.
Awesome. Well, folks, if you have any other questions, feel free to drop them in the chat, but we're going to move on to the next speaker.